It is my pleasure to introduce you Drs. Irving Vega, who is one of our speakers for the Neuroscience Society series. And the title of his talk is Social and Biological Intersects in Alzheimer's Disease, Deconstructing Racism. And I want to talk a little bit about Dr. Vega, which I know him for quite a long time now. And he obtained his undergraduate degree in biology from the University of Puerto Rico, Mayagüez campus. And followed by that, he continued his research training at the Department of Cell Biology and Neuroscience at the Graduate School of New Brunswick at Rutgers University, where he received his PhD. Dr. Vega also completed a postdoctoral fellowship in neuroscience at the Department of Neuroscience at Mayo Clinic in Jacksonville, where he developed his research career focusing on the pathobiology of Alzheimer's disease. And then after that postdoc, he joined us here at the University of Puerto Rico, Rio Piedras, and finally he moved in 2014 to the Department of Translational Neuroscience at Michigan State University College of Human Medicine campus in Grand Rapids. So here at MSU, his research focused on molecular and biochemical mechanisms that modulate the accumulation of pathological tau proteins in Alzheimer's disease and related dementias. And Dr. Vega is also working on ethnic disparities and the influence of ethno-racial factors on blood biomarkers in Alzheimer's disease. So obviously this is a very important topic. We're so proud to have Irving again with us. And I want to mention that Irving was really the mastermind of our Neuro-ID program many years ago. And we really appreciate him coming back to contributing to us and making this a very, very successful program. Thank you, Irving, for being here. And we look forward to your talk.
Thank you, Carmen, and thank you for the invitation to you and Jose Enrique. It's really an honor to be here with you guys. There are students here in the room, and there are also, I see few connected through Zoom. Today I'm going to talk about social and biological intersects, something that when we try to study diseases, we have to keep in mind how the environment affects the biology and how that environment contributes to the risk of developing different diseases and how that actually creates the differential environment, creates the aspects that we call or refer to as health disparities, how some groups are actually more at risk than others to develop certain diseases. And it sometimes can get confused with biological risk, taking or not taking in consideration the social context and the contextual factors where that biology actually has been developed 2 years. So it's very important for you guys, the new generation of scientists, to always maintain the aspect of contextual factors and the intersect between the environment and the biology. So before I continue, I don't have any financial conflict of interest to declare. These are some of the roles that I play in different organizations. So the opinions that I made today don't necessarily correspond or are represent the views of these organizations that I'm associated with. So I'm going to start with a brief introduction about Alzheimer's disease, and then we will move slowly about what I was talking regarding contextual factors and how we try to dissect the contextual factors from the biology and using that information to better understand the progression of Alzheimer's disease. Alzheimer's disease was first described by Alois Alzheimer, a neurologist, pathologist that worked at this psychiatric hospital in Germany, where Auguste Dieter was actually institutionalized by her husband because he couldn't deal with her anymore. And Alois Alzheimer started looking at her by the end of the 19th century, 1896, 1895. And a famous phrase was that she mentioned in one of the interviews with Alois Alzheimer was, "I have lost myself." He developed a very good interest on her because sometimes when he was interviewing her, she was actually very normal, not to have any kind of disease presentation. But then during that afternoon or the next day, he was a complete different person. So he immediately identified that case as something different than the other psychiatric patients that he was dealing with at this hospital. When she passed away, he performed an autopsy and started studying the brain and using different techniques to stain the brain. At that time was developed by Golgi and others, the silver staining technology that used actual silver to stain and look at the brain histologically. He was able to identify two particular lesions in the brain of the patient that we're gonna talk in a second. But Alzheimer's disease is actually currently from that 1896 to 1903, 1902, when he presented the case of the Agosti Dieter. It started after that presentation, people start referring to the disease as the disease that Alzheimer described, and then his actually mentor decided to call it Alzheimer disease. Currently, there is about 6.5 million people diagnosed with Alzheimer disease in the United States, including Puerto Rico. And it's about 1/3 per age group, 65 or older, to 65 to 74, 75 to 84, and older than 85. It's about 1/3 of the presentation in those age groups. I want to point out that here is start at 65 years of age, this graph, but there are cases of Alzheimer's disease that are earlier than that. And those are called early onset Alzheimer's disease. And in a second, I'm gonna talk about that. There is, in these 6.5 million, there is demographically an overrepresentation of Blacks and Latinos. So African American Blacks are 2 times more at risk of developing Alzheimer's disease than any other ethnic groups. Latinos are 1.5. And it's actually a disease that we still don't understand why, but it has some sex as a biological variable, where women are 2 times more at risk of developing Alzheimer's disease than males. And currently, about 60 to 65% of the Alzheimer's cases are females. So there is more representation, and there's a lot of hypotheses about that. That if you have questions, we can talk about that in the question and answer. So in here, illustrate. This is a report from the Alzheimer Association. Every year they release what they call the facts and figure, and illustrates how healthcare, and this is an aspect of the racial disparities associated with Alzheimer's disease. And these numbers illustrate how, in terms of the need for diversity in healthcare, where the patients were asked whether it was important for them, for Alzheimer or dementia healthcare provider to understand the ethnic background. And you can see that for most of them, it was 85% for Hispanic, Latinos, 89% for Blacks, Americans, African Americans, 84% for Asian Americans, and Native Americans were 92%. So for the underrepresented ethnic groups, it was very important that it was taking in consideration the ethnic and racial background when was taking in the healthcare providers for Alzheimer and dementia. At the same time, the other question was, how confident are you that patients currently have access to providers who understand their ethnic or racial background? As you can see, again, the vast majority, especially for Hispanics and African Americans, African American was the worst with Native Americans, that they were not confident that they have providers that understand the ethnic background. As I mentioned at the beginning, that's why you as a future scientist is important to understand the contextual factors, because that environment dictates not only the risk, but also the progression of the disease. What you can see in this one is the percentage that feel discriminated when they're seeking healthcare. And again, when you look at the whites, of course, they don't feel that discriminated when you compare to the other underrepresented groups. Something important that to illustrate here is the barrier for this underrepresented group to access healthcare early on, because what we're gonna see, there are already information that can be used to reduce the risk of the individuals to develop Alzheimer's disease. But the information and the care needed to actually reduce that risk is not that accessible. And one of the aspects is the perception and actually suffering discrimination. Blacks, African Americans, and Latinos are also more likely to not receive a diagnosis of Alzheimer's disease or dementia among the ethnic groups as well. So some people attributed that the access that they have is primary care physicians like internal medicine or family medicine who are not equipped to diagnose cognitive impairment or willing to give that diagnosis. And then the access to neurologists, depending on the health insurance, if they have health insurance, then it's almost impossible. Right now, there is a shortage of neurologists in the United States. And the first-time visit for someone to visit a neurologist could be up to between 6 and 8 months of wait time. That is with insurance. Imagine someone that is on Medicaid or don't have health insurance. It's almost impossible to get access to a neurologist. And that is in the United States, okay? So what Alzheimer found and described in the early 20th century was the accumulation of two pathological hallmarks. One in the extracellular space, in the parenchyma, that is the accumulation of a peptide of 40 or 42 amino acids that form these plaques called amyloid plaque or senile plaques. And the other pathological hallmark that was found is actually inside the neurons, and is called neurofibrillary tangles, and is the aggregation of a protein known as tau. And the best known function of this protein tau is the stabilization of the microtubules. However, there are other functions that are attributed to this tau protein, from signaling to synaptic transmission to vesicle trafficking to even chromosome stability, because it can be found also inside the nuclei. So the tau protein gets aggregated intraneuronally, especially in the somatodendric compartment, and the amyloid peptides aggregates as plaques in the extracellular space. So that is actually what neuropathologically describe or define or diagnose a case of Alzheimer's disease. So the diagnosis of Alzheimer's disease is postmortem, it's the confirmation. What you get at the clinic is possible or probable Alzheimer's disease. Probable Alzheimer's disease is when someone has a clinical presentation that is very typical, that start with a chief complaint of memory problems and executive function, meaning taking decisions, like finding, forgetting how to pay the bills, or giving donations to someone that asked for a donation, and then they give $1,000 when they have 500 in their banking account. So that aspect of decision-making and memory are the chief complaint. That's when it's diagnosed as probable. Other times, and most of the time, the chief complaint is sometimes outside of the memory of, aspect, and it usually is either behavior that the person is suffering from anxiety or depression, or the person has problems finding locations. Like this individual was every week used to go to do grocery shopping at this particular supermarket, and now it reached the supermarket, but it cannot return to his or her home because got lost. And that's the spatial memory, visual spatial memory is affected. So when it's behavior problem, is because from the temporal lobe, especially in the area of the hippocampus and the entorhinal cortex, the limbic system, is where the pathology usually originates, and then emanates into the frontal lobe. When it goes to the frontal lobe, then the clinical presentation is associated with memory and behavior. When it goes to the parietal lobe, then it's the visual spatial deficiency or impairment. So even though this illustrates a hierarchy going first to the frontal lobe, we know now that it can go actually backwards toward the parietal lobe first, and then the frontal lobe. So there, depending the individual, it can go first to the frontal lobe or the parietal lobe. But then as the disease progresses, the pathology continue through the cortex. This is a cortical disease. It start emanating two different cortex, temporal, parietal, occipital. Even in the occipital lobe we can detect this pathological hallmark, where the cerebellum is completely spared. So the cerebellum you cannot see any pathology in the cerebellum. We usually use the cerebellum as a control in our experiments when we are studying postmortem tissue. So this progression of the pathology and the presence of these pathological hallmarks led to the main or predominant hypothesis in Alzheimer's disease, which I think it has become dogmatic and has been the reason why the study of the disease has stalled, and all the clinical trials directed to the reduction of the amyloid plaques have failed. And it's time for us to start thinking outside of the amyloid cascade hypothesis, but this is the most prominent one, and I will tell you why it's more prominent. The peptide of 40 or 42 amino acids that I mentioned that form these plaques come from a protein known as amyloid precursor protein, and it's a integrated membrane protein, which the biological function is not well understood, but apparently the protein might be involved in neuronal development and also in synaptic transmission, but the actual biological function is not well understood. This protein is endocytosed, internalized, cleaved, and then these peptides go back to the membrane in a vesicle and are released to the extracellular space, okay? And once those peptides, usually the one that is released is the 40 amino acid, long peptide, but when the 42 amino acids start being released, that one is prone to aggregation and brings the whatever 40 amino acid peptide is there, and then they form these aggregates in the extracellular space. They are familial or inherited mutations associated with the amyloid precursor protein. Actually, these mutations were found studying Down syndrome cases. Well, not a mutation, but the APP. In Down syndrome, about the late 30s, mid 40s, they are more prone to develop dementia type of symptoms. when they donated the brain to study, they found that they had the similar amyloid plaques in the brain of individuals with Down syndrome. Down syndrome is a trisomy of the chromosome 21, right? So then they say, okay, this is the same peptide that is found in Alzheimer disease. And the cause of Down syndrome is the trisomy of chromosome 21. So if there is a gene associated to Alzheimer disease, must be then in chromosome 21. So it starts sequencing, and then they found mutations in the chromosome 21 in the gene that encode the amyloid precursor protein. And those were the first mutations identified associated with inherited Alzheimer disease. They have been found, and I have a slide later on to talk more about that. They have found mutations on proteins called presenilin 1 and presenilin 2 that cleave APP, that are the responsible to cleave the APP and produce those 40 or 42 amino acid long peptides. And they have been found mutations in those genes that the individuals that inherit those mutations is autosomal dominant, and they end up developing Alzheimer's disease early age. But there are cases that have been reported in their 40s with a diagnosis of Alzheimer's disease. And when they are individuals that are developing dementia type symptoms in their 40s or 50s, it's actually suspected that they have a mutation in one of these three genes, either APP, presenilin 1, or presenilin 2. The accumulation of the peptides led to cellular stress that activate different signaling pathways. And these signaling pathways then have increased post-translational modifications on the tau protein. Once the tau protein becomes highly modified, aberrantly post-translational modified, it becomes aggregated. It starts to form aggregates. And those lead to the formation of paired helical filament, or PHF, which then collapse into the tangles and then leads to the neurofibrillary tangles. The accumulation of those paired helical filament, and before that, the oligomers, small filaments, is actually what is toxic to the neurons, not the tangles. The tangles and the plaques actually now we understand that are neuroprotective. It's a way for the cell to take and reduce the entropy that these oligomers and these filaments cause inside the cell or outside in the extracellular space, for example, done by amyloid peptides or inside by tau. So the accumulation of these plaques that Alois Alzheimer identified are actually now referred as less toxic molecules than the oligomers and the star filaments that A beta form. Those are the ones that lead to neuronal dysfunction and then death. It's important to highlight here that it's through tau that amyloid induces the toxicity. If you don't have tau, and you only have aggregation of A beta, there is no toxicity to the neurons. The neurons don't die, okay? There is some synaptic dysfunction, but not to the level to develop some kind of a presentation of cognitive impairment. And these studies are mainly done, obviously, in animal models. So the toxicity is well understood that is through tau. And why is that? Well, tau, you saw that figure at the beginning where I show you the progression of the disease from the pterygium cortex to the hippocampus, then it goes to the cortex in the temporal lobe, and then move to the parietal or the temporal, so on. So what we understand now is that the oligomers of tau protein, or small filaments of tau proteins, can be transmitted from one neuron to the other one, okay? And some people wrongly call it prion-like transmission. And that created a lot of confusion because the prion disease is an infectious disease that is mainly based or completely based on the transmission of this protein, that if you consume that protein either by meat or contact with blood that someone had the disease, that prion protein touch your prion protein and then transform the conformation, and you have the aggregation, and you develop very rapid dementia, type of dementia, and then you die in about 5 years. So prion disease is a very infectious and aggressive type of dementia. And the people, because it transmits from neuron to neuron, okay? And some scientists start referring a tau transmission, and actually it's alpha-synuclein in Parkinson's disease. We found that, people found that that happens too, because it transmits from neuron to neuron. They start calling it a prion-like transmission. And that was a mistake, because now people start thinking that tau is infectious, right? And it's not that the case. There have been several studies of people with Alzheimer disease that have donated blood, and people have looked at it, and people that receive that blood, and there's not any higher risk than the normal of those individuals to develop any type of dementia. So is the evidence so far indicates that tau is not like a prion protein, it's not infectious. But what we know, we start to understand, is that definitely tau neurons can uptake oligomers and small filaments of tau, but not the tangles. If you have the tangles and you take an animal and you inject the tangles stay outside the neurons. If you now sonicate those tangles and develop a small filaments or oligomers, the cells can uptake that. And when the cell uptake those oligomers, it somehow gets released in the intracellular space. And I want to emphasize somehow gets released in the intracellular space and touches other tau proteins. And that serve as a nucleation factor for the tau now that was inside those neurons to become aggregated. And then the system continue replicating. Now you have more oligomers that get out of that neurons and go transmit to another neuron, and so on and so on. And what it has been shown is that that transmission takes place with neurons that are highly connected. So that's why you see that progression and cannot pronounce the word. It goes in that transmission in areas that are really connected, right? So it has to go emanate from one region and continue transmitting the pathological, and it is a requirement that they are connected, right? So the hypotheses are that the local transmission can take place transsynaptically, meaning that the proteins are connected, and to the post and presynaptic terminals, that transmission the vesicles are passed through the synaptic cleft. The other hypothesis is that it can go into these nanotubes that are formed between neurons to where information from the cytosol is passed from one neuron to another one, or that they're actually secreted locally and are uptake by the neighboring neurons, okay? There is a huge debate on how that takes place. So the accumulation of these pathological hallmarks, the oligomers, the small filaments, and the tangles and amyloid plaque are actually used right now as biomarkers to see the progression of the disease, to monitor the progression of the disease. So here I'm showing a graph of the correlation that try to simulate the correlation between the accumulation of the amyloid peptides and tau. And that is done by analyzing the CSF, cerebral fluid, spinal fluid, or either by brain imaging using PET scan. There are radioisotopes right now that are specific to A-beta aggregates in the brain or tau aggregates in the brain. And even though there's still controversy that it can be cross binding for the different aggregates, they're right now very specific to differentiate between A-beta and tau. So these biomarkers are actually accumulating through time as the individuals age. And it gets to a point where those, a threshold point where those individuals that have accumulation of these aggregates then lead to mild cognitive impairment, and that mild cognitive impairment then move toward full-blown dementia. Okay, but not everyone that develop MCI or mild cognitive impairment transition to dementia. Actually, about less than 50% of individuals that are diagnosed with mild cognitive impairment that transition to dementia symptoms. Okay, and dementia is an umbrella term that is a syndrome that includes all behavior, cognitive problems, and is the term referred because having a pure Alzheimer disease is very difficult. You always have some vascular or other pathologies in the brain. It's not as simple as A beta and tau. Okay, it's more complicated than that. But the point that I want to make with this specific slide is that you can see the transition and accumulation of these pathological hallmarks. What it means is that you can have these pathological hallmarks in the brain. Actually, I can have those pathological hallmarks right now. It doesn't mean that I'm going to develop the disease. We still don't understand why is that threshold that some individuals transition and will present symptoms associated with the accumulation of these toxic molecules, and other people will die completely normal, with normal aging, with similar amount of pathological burden of these pathological molecules without any presentation of symptoms. So another myth that I want to give you to resolve today to you guys is that Alzheimer's disease is not a disease of all people. It presents in all individuals because of different factors, including cognitive reserve and different the environment and how the individual is still engaged in social activities, in exercise, the contextual factor, the eating, the healthy diet, etc. But we know now that the pathological hallmarks that I just described can start accumulated in the individuals as early as their late 30s, maybe in their 40s, okay? And it can be 20 to 30 years before clinical presentation, okay? So it's a disease that starts in about 30 years of age. I mean, the pathological hallmarks start accumulating, but then some individuals will have clinical presentation, other ones not. And I would argue that most people would not develop clinical presentation, and the minority is the one that develop or present the clinical presentation. So again, it's not a disease of all people. It's a disease that presents in all people. So we can do a lot of things before we get to our 65 years of older, or even when we pass the 65 years of age, to reduce our risk of clinical presentation, okay? So because of the dogmatic view of Alzheimer's disease with the amyloid cascade hypothesis, there is this platform known as the Atm Framework, okay? And the Atm Framework is actually, Atm stands because of the amyloid, the A, the T is tau, and the N is neurodegeneration. And they're biomarkers that are used to detect neuroinflammation or inflammatory response. NFL or neurofilament light protein, or GFAP, okay? And these two proteins are known to be associated with inflammatory response in the brain. And they are very specific to detect brain injury. They're not specific, they're very sensitive to detect a brain injury, but not specific to determine whether you have Alzheimer's disease, Parkinson, or even traumatic brain injury. Individuals that have concussions, when you look at the blood or CSF, you will have NFL or GFAP in the blood, similar to someone that has Alzheimer's disease. So it's not they're not specific, but they're sensitive enough to at least say there is a problem in the brain, okay? So that is the N. So A for amyloid and T for tau. So the Atm framework is whether you have detection of these biomarkers. So you can, as I mentioned before, you can detect amyloid. In this case, is the Pittsburgh compound B, was developed in Pittsburgh. So that's why they put it at the University of Pittsburgh and in collaboration with UPenn. And they developed this compound, the first one, to actually specifically detect the accumulation of A beta. So the red here indicates A beta aggregations in the brain of the individual that have Alzheimer-like dementia. Another way to detect is lack of glucose consumption or consumption of glucose in a specific brain regions. So you can see the correlation. Red here indicates A beta. A reduction of red indicates less cell activity, right? So there is a correlation between reduction of cell activity and accumulation of A beta, meaning the neurons are dying, okay? In this individual with Alzheimer type dementia versus a normal aging individual. So the accumulation or detection of amyloid, in this case, or glucose activity, neuroinflammation, but also tau in the CSF or in the brain imaging lead to this Atm framework. Something that I want to mention before I continue is that the increase of tau in either in the CSF or plasma is associated with Alzheimer-like pathology or TBI, or there is an injury in the brain. The reduction of detection of A beta in the CSF or the plasma is indicative of Alzheimer type dementia, the reduction. And you say, why is that? Well, the thing is that the A beta, when it aggregates, cannot be filtered out of the brain, and then it gets trapped inside the parenchyma, and then you detect the reduction of the A beta peptides in the plasma of CSF, meaning that the reduction implies or is a proxy for aggregation of that peptide inside the parenchyma. So that's why you look for reduction of A beta as a diagnosis for Alzheimer-like dementia, okay? But then tau gets released. Remember, tau is the one that is transmitted, gets released, and then the increase of that release is the one that is detected in the CSF and the plasma, okay? Good. So a little bit of genetics to start going toward, now we're gonna move toward the health disparities aspect. But before, we have to talk about a little bit about the genetics of Alzheimer's disease. So I mentioned that mutations on APP, presenilin 1 and presenilin 2 are autosomal dominant, and individuals with this mutation are most likely to develop Alzheimer's disease. Although there are mutations in presenilin 1 and APP that are actually protective, but we're not going to talk about that today. But they have mutations, especially you can see family history, not jumping any generation. You will see individuals that will have the propensity of early onset Alzheimer's disease. But there are other genes that have been found in genetic association studies that are they're common in the population, but they have a low risk of developing Alzheimer's disease. But the accumulation of those variants, SNPs, specific SNPs of different genes, then the accumulation of those then will dictate your risk of developing Alzheimer's disease. There is another gene that is ApoE4. You have ApoE2, ApoE3, and ApoE4, epsilon 4. And if you have 2 copies of ApoE4, meaning you're homozygous for ApoE4, you have 50% chance of developing Alzheimer's disease, higher than anyone in the population. But don't worry, that's only for whites. It doesn't apply for Latinos and African Americans and Black. It's mainly on non-Latinos or non-Hispanic whites. The correlation with Blacks and Latinos is not that strong. but those is known as have a medium risk. And then, you know, the more accumulation of these SNPs, you have more probability of developing Alzheimer's disease, have more risk. In a summary, when all these genes are studied, they tend to fall in three different major biological processes. Obviously, inflammation, lipid metabolism, and endocytosis. And these are three areas of intensive study, especially inflammation. There's some people that indicate that inflammation actually is the etiology of Alzheimer's disease. So you start with because of all the accumulation of injuries and even COVID-19 now, which induces inflammation in the brain, you can develop these inflammatory response that then goes into this vicious cycle and then continue killing your cognitive reserve. And then you try to repair, and it's like a saving account that you keep taking things out, and then you get it to a point that now you don't have any more savings, and now any lesion leads to more, you be more closer to clinical presentation. So something that had been found recently, and this is a paper published last year, that people start looking, okay, is there any biological relevance about to explain or to justify why African Americans or Blacks are more at risk of developing Alzheimer's disease? So they were start looking at, and they found a particular set of genes that are associated with higher risk of developing Alzheimer's disease in the African American individuals. And, you know, this is obviously very controversial because first, the cohort that was studied, and also because how those mutations or how those changes take place to different generations, whether that actually is the factor that contribute to Alzheimer's disease. In order to make that, you have to then take those individuals and put it in the same environment that other people that have lower risk, and then see if they still develop Alzheimer's disease. So anyway, so they found those mutations. We also found mutations that migrate or are actually specific to Puerto Ricans. A mutation in presenilin-1, a change in glycine to alanine in the presenilin-1 gene is actually associated with Alzheimer's disease in a cohort of Puerto Ricans. They're here in Puerto Rico, in Washington Heights, in Philadelphia, and now recently was also validated in Miami. And what they have in common is that they're all of Puerto Rican descent. So that is the Puerto Rican mutation for presenilin-1 that is inherited. It definitely is associated with developing early onset Alzheimer's disease. And these individuals have a more rapid progression and aggressive progression with a diagnosis to death between 5 to 10 years. However, having a mutation of presenilin-1 don't necessarily dictate whether you're going to develop Alzheimer's disease or not. And that was actually a study done with a group in Colombia. These individuals are followed by researchers at Harvard Medical School in Colombia. And this family have different generations, and they found this mutation on mutation in presenilin-1. But then they found this individual that even though has the presenilin-1 mutation, and members in her family were developing symptoms at late 40s, in their late 40s. This individual is already 72 years old and only with mild cognitive impairment. It's an N of 1, okay? And that is the power of an N of 1. And they say, why this is an outlier? It has the same mutation. What the heck happened? So this individual has a second mutation, in the ApoE3 that is called the Christchurch mutation. And this individual is homozygous for this mutation. And it seems that having this mutation is protective. So even though she has the mutation in the preceding one, and every other member of the family have developed Alzheimer's type dementia in their late 40s, she's already at 72, 73 years of age and is still cognitive impaired, not full-blown Alzheimer's disease. So of course, the story now goes to try to find out what is the relationship with ApoE. And remember the three biological pathways, lipid metabolism. So what is known about this protein, ApoE, apolipoprotein, is more related to lipid metabolism. However, it is, as we understand now, there's other functions beyond the lipid metabolism and probably involved in synaptic transmission as well. And regulation of long-term potentiation by delivering cholesterol and affecting the membrane of the neurons, and in that way affects the action potential and the polarization and depolarization of the membranes of the neurons, the neuronal membrane. So this is a story that is really interesting and is still ongoing. And I think we have a Puerto Rican here in the authors. Let me see, where is it? Ed Marie was part of this study. And Marie Guzman right here. A student that actually did the undergrad here in Rio Piedras, and she's a neuropsychologist, and she worked now, she's doing, she's at this point, she was a postdoctoral fellow, but now she's an assistant professor at Harvard Medical School. She has contributed a lot in the characterization of this cohort, the Columbia cohort, working with Jaquel Quiroz in Harvard. So, okay, it got stuck. Come on. It got too excited about that now got stuck. Let me just exit for a second and come back. Okay, sorry. Let me go back. And share. Come on. Okay, let me see if now we are, okay, we're moving. This slide is to show you that there are other mutations other than presenilin-1, presenilin-2, and APP that are induced early onset Alzheimer's disease, and there is inherited, is in the families. And this is a mutation in MTCO2 that is a mitochondrial protein. And these individuals, as you can see, is their mid-40s, late 40s. They have mutation in this gene, different type of mutations, and they develop Alzheimer type dementia. But there are others individuals with the same mutation in the same gene that develop sort of a normal age of onset of Alzheimer type dementia. And some of them have family history, other ones don't have family history. So this could be a novel mutation, or there are other genes in these individuals that now develop them, make them at risk of developing Alzheimer's disease. Well, we don't know. But I just want to mention that there are other genes other than APP, presenilin-1, presenilin-2, but they are rare. Looking at biomarkers, especially in the peripheral blood, that there is the interest of identifying biomarkers that help increase the specificity to diagnose Alzheimer's disease and the sensitivity, but mostly the specificity. People are starting looking at race as a biological variable. And that's where I have problems. This study was published, the people was you. And it's, for me, creates some of the implicit biases that are among researchers in the Alzheimer's disease field. And I don't have any problem saying this because I said even in study sessions and different conferences. And I point them with name because I like to do that. And these individuals have definitely, their research illustrate their implicit bias. So I want to drive your attention to this statement here. They say that there's research finding biomarkers that are differentially detected in blacks versus white. They concluded racial differences in Alzheimer biomarkers suggest possible race-dependent biological mechanisms that contribute to expression of disease, end of quote. So nothing about contextual factors, nothing about other types of environmental factors that may contribute to the disease. They are indicating that there is a biological mechanism, and that is no evidence in their paper about that. Only that they found that the typical biomarkers of A beta and tau, particularly tau, is less, they detect less tau, but more hippocampal atrophy in blacks versus white. So white have more tau and phosphorylated tau in plasma and CSF than blacks. But then the blacks have more hippocampal atrophy. And that is already, this was a 2019 paper, the beginning of 2019, but that is already been completely shut down because it's all about selection bias, right? Where you select your individuals, you have to define the aspects of that selection. Where are those individuals, the social demographics of them, what is if it's known that there's social stress in that area, you can now, by zip code, you can determine the social stress of specific areas in the United States. You can determine whether pollutants are in that area. So there are different factors that you can better define your cohort and put it in context, right? It doesn't mean that their data is wrong, it's that it's out of context, trying to generalize something that is not they cannot generalize. And number two, attributing to a biological aspect when they don't take in consideration the social aspect. So this actually is a very comprehensive review that just came out at the end of 2021, and it actually goes into those aspects. So when you have Alzheimer disease centers, that's the ADC, they are usually a hospital with a very centric position in the city where they selectively recruit people that have access to those hospitals, right? People that can go to those hospitals. So that usually defines as urban regions, people that are socioeconomically a little bit better than people that are in rural areas or actually in areas that are even in the urban area, but they're with a lot of social stress. When they look at that, they found that yes, they were validated that amyloid and tau, there were differences. But when they look at the positions in the brain of amyloid and tau, there was no difference. And when they started comparing other studies where their selection was done in a different way, they tried to identify, especially for PET scanning, that there were some diverging in the direction where higher, lower accumulation in the cortex of amyloid plaques in Black individual versus white. So what that says is that depending on the individual that you're studying, you want to find some differences. So it's not just because you're Black, not because you're white, but where you are selecting this individual. And the same thing has happened with tau. How they have found even in Latinos, some differences in the accumulation of tau. For Latinos have been found a larger, more than white, and for Blacks have been found less. But then when you look at tau, for example, but then when they look at the different studies and try to put them together, there's no difference between them. And more recently, it have been found, especially studying Mexicans in South Florida, in South Texas. They found that specific comorbidities dictate whether you can detect this amyloid or tau in the plasma. So they look at this hyperlipidemia, they look at hypertension, they look at diabetes, and how those chronic kidney disease. Because of course, if you have problem in your kidneys, how you eliminate these peptides from the blood, it gets affected. So depending on these comorbidities will be the amount of total tau of A beta that you will detect in the plasma. So not only the social aspect, but also the comorbidities, the diseases that are very prevalent in Alzheimer's disease patients, you have to take in consideration in order to detect these biomarkers in the blood. In the case of ethno-racial differences, this is a study done in UPenn, and actually when I was here at the University of Puerto Rico, we collaborated and we found that that the age of onset of the individuals there, Latinos, that 85% of that cohort were Puerto Ricans, was 68 years of age compared to 73 for Black African Americans and 72 for non-Hispanic whites. So it was like early onset compared to those individuals. And also the cognitive impairment was actually more severe in these individuals than Blacks versus non-Hispanic whites. So the disease progression was actually faster in those individuals, and the education was significantly lower, was about the 6th grade versus high school for the other ethnic groups. So there is education as a proxy for many other things, right? And the progression was actually more associated with a higher degree of depression and well, in this case, this is the index of depression. So when we were looking that 85% of that cohort were Puerto Rican, we say, oh, you know, if it's because of Puerto Ricans, let's see what happened in Puerto Rico. So what we found was that when we look at mortality rate, the mortality rate, white here means low mortality rate, and dark black means high mortality rate. So in the metro area, Maya West area, the mortality rate is in comparison lower, associated with Alzheimer's disease versus Arecibo, Aguadilla, and the area of Caguas, that region there. So those have significantly higher mortality rate, even higher than the Puerto Ricans in the United States. Versus in metro area, Maya West, that we have lower mortality rate associated with Alzheimer's disease. So already questioning that just because you're Puerto Rican doesn't mean that you have a higher risk of Alzheimer's disease. It's not just because you're Puerto Rican. So then we look at the record and did a retrospective study and found that the age of diagnosis, it was 70, in all regions, the average was 77 compared to 68 in Philadelphia. Again, illustrating that just because you're Puerto Rican doesn't mean that you are having an early onset of Alzheimer's disease versus other ethnic groups. There are other, what this is pointing out that there is other factors. And here we see the education in there was 6th grade, in here in Puerto Rico is high school diploma, right? So there's some differences there that is we cannot explain just because of ethnicity or education. And again, the initial study test of cognitive performance, it was similar to the whites there in Philadelphia, not as severe as the Puerto Ricans in Philadelphia. So that start thinking about what is happening in that area. In more recent study, we found that we started studying using this large data set that is nationally represented of Latinos in the United States. And we say, okay, if it's Latinos, just being Latinos, a proxy for developing cognitive impairment, let's look at the Latinos in the United States. And what we found is that if you are US-born Latino, that's what USBL means, you definitely, even after all the corrections. in Zoom? I'm frozen? Yeah. Oh, okay. Let me stop again. Did it stop the presentation? Okay. Let's share again. Let's go here. Let's share. Better? Okay, thank you. Sorry about that, those that are connected to Zoom. So let's go back. Oh, and now here I'm frozen. Oh, okay. Oh, completely escape. Everything is frozen now here. Oh, yeah. I cannot see even my, it's moving in here, but okay, let me just do this stuff there. Okay. Okay. Mm-hmm. It's stuck. Okay, let's try again. Okay, you can see that. This. Okay, presentation. Come on, work. Okay, good. Okay, I don't know, change. I mean, oh, okay, now it did. There is a little bit of delay. Okay, let's see if moving forward. Okay, good.
Yes, it's moving forward. It's just that it wasn't continuing the slide, so that's why I was. But now it's moving. Okay, it's fine. Just continue, even though it's not, you know, in presentation mode.
Okay. Okay, good. So what we found was that looking at that large data set that is naturally represented of Latinos, we found that US-born Latinos at baseline have worse cognitive performance than non-Latinos white after correction by age, sex, education, etc. Their cognitive function was worse than than non-Latinos white. However, when we stratify that and look at Latinos in the United States that migrated to the United States before 18 years of age, between 18 and 34 years of age, and 35 or older, their cognitive function was similar to white, no statistical different to white. So again, US-born Latinos are worse in cognitive performance, and Latinos that were migrated, regardless of the age, perform at the level of white. So it's something about Latinos born in the United States that actually are more at risk of developing cognitive impairment than those that are migrating. And that goes against all the political issues and questions that migrating are destroying the United States and all that. Actually are the US-born Latinos that are performing worse. And that is true by any domain. We look at memory and also sequential. That is actually the serial 7 is more about that sequential analysis and processing of in the brain. Regardless of the domain, we found that those individuals that are US born always perform worse than the migrants. Although in processing, those individuals that are migrated older or late in life, they were a little bit more cognitive impaired in the domain of processing. And that could be because of language issue on the essay than actual processing problems. because in the other domain of memory, they are actually performing better than the younger individuals that migrated younger to the United States. So what these point out, and everything that I've been building up to this point, is that there is a vicious cycle of inequality, and that is what we call social determinants of health. So how the contextual factors, meaning the neighborhood where they live, the quality of education, not the years of education, but the quality of education, the social stress in those areas where they live actually affects education, which then affects health and affects access to healthcare, okay? So the socioeconomic factor is very important because that dictates whether you have good education or you dictate or good healthcare, okay? And as you probably know, in the United States, the quality of education is directly attached to the neighborhood that you live in. The better the neighborhood and more rich is the neighborhood, the better the public school, okay? So that contextual factor contribute to your education and contribute to your healthcare, and that's a vicious cycle that people sometimes don't leave. And what NIH have developed is what is known as the from the National Institute on Aging is known as the Health Disparity Research Framework, which is, in other words, how the social components influence the biology of the individuals. And it's divided in these four major components: environment, sociocultural, behavioral, and then the biology, okay? And in a better representation is this actually extracted from this review published in Alzheimer and Dementia, where they look at the environmental factors, they look at sociocultural factors, again, coping mechanisms, smoking, physical activity, depression, the social safety net, meaning that social stress that is in those neighborhoods, access to quality of education, the environmental factors that go from a structural racism. Remember that Blacks and Latinos in the United States, when they go to healthcare, they have this perception of being discriminated. The neighborhood, the pollution, access to healthy diet, okay, which is attached to also the socioeconomic factor. A stressful life event, and that is tied to the coping mechanism, and how all this then influence to the biologic aspects. In the case, for example, inflammation, metabolic disorders. So remember that I was telling you about diabetes, hyperlipidemia, kidney dysfunction, but in the case of diabetes and all the metabolic disorders, is associated with a higher risk of developing dementia. And in which groups these metabolic disorders are more present? Latinos and Blacks. And why that? Is because of the diet, the stress, etc. Okay? So all those aspects of environmental factor and socio-cultural factors have an incidence on the biology of the individual. And I'm not talking about in here, it's because it's a large area study, epigenetics, how these environmental factors affects the epigenetic code of the individuals and whether that is transmitted to the next generation. Those epigenetic changes are transmitted to the next generation, making them following the generation, even if they move out of these areas, they still mark to develop or be a higher risk of developing dementia. And then all that leads to accelerated aging, brain aging, and the propensity of developing these pathological hallmarks, again, the Atm framework. Unfortunately, what we know, and I say unfortunately, is because that information is not transmitted to the community, that the consensus is that if we deal with all these social and environmental factors, we can reduce at least half of the dementia cases. Half. So instead of 6.5 million, we can have about 3.5 cases, okay? Just by dealing with the risk factors, okay, by bringing that communication to the individuals and dealing with the social, cultural factors and environmental factors. So there is ongoing work that we are doing and others to bring that information to the community, to make it accessible, to build bridges between community-based organization and community-based clinic that are the clinics that these individuals have more access to. And in that way, by empowering, although I don't like to use that word, but by bringing resources to these community clinics and community-based organizations, we can reduce that gap between knowledge and practice. And the individuals can use that information to make better decisions for their healthcare and also find ways to reduce stress and eat better, etc. So that's the message that I have for you today, and then I will be happy to answer any questions you may have. Thank you. Okay, questions? Sure.
Is there like a proper diagnosis or clinical study that we can go to verify we have that mutation? Because I was in my family, we have a history of Alzheimer, and we've had two different cases. Like my great grandma, she had Alzheimer, but she died at like 97, so really old age, and she started Alzheimer at like 90. But my grandma, she started Alzheimer at like late 40s, so it was really different, and she's now 60, and she's just another person. We don't really remember her, and she doesn't remember anyone. She's already in bed, and we don't really give her many years. And yeah, like now I'm worried because I've always got told that Alzheimer is not really. Does not really mean anything if your family history has to do with anything, but now after this presentation, I'm like,
well,
maybe my mother should get checked.
For the benefit of the people in Zoom, the question is whether there is a diagnostic test for inherited Alzheimer disease, summarizing what you said. And there is. You can test for these three genes, APP, presenilin 1, and presenilin 2. However, it poses an ethical question to the clinicians because is information can help or can harm, right? Because there's no cure or available treatment for Alzheimer's disease. So it poses a very ethical question to the clinicians when a patient asks about genetic testing, okay? And there are genetic counselors that are the ones that are referred to in those cases in order to have the psychological aspect and try to prepare the people for the potential positive identification of a mutation that is associated with inherited Alzheimer's disease. And how you're going to deal with that information. And then when that genetic counselor is secure that you are well, you're going to be okay, then the process continues. If you have the resources to do that, because most, if not all health insurance pay for that kind of test. So most cases is an out-of-pocket kind of expense. So you have to pay for the whole thing. The health insurance will not pay for that. So again, it poses a very ethical question about how you're gonna deal with the information.
So when you do like tests like 23andMe and they send you're prone to have this problem. Do they take into account the Alzheimer's?
Yeah, they're looking for APP. They look at presenilin 1, presenilin 2, alpha-synuclein, progranulin. They're looking at all these genes, and yeah, what they are doing with this information is what we are concerned about. Yeah, and how, you know, the confidentiality and how, you know, you process that information and all that is another ethical issue there. Yeah. But for example, the Alzheimer Association say that the benefit outweighed the potential harm, because then you can prepare and make informed decisions about how you want to be taken care of, what are, you know, your inheritance, how you're going to distribute it, what you want to do, and make a lot of decisions to take the burden away from your family members to making decisions for you. And that's Alzheimer Association, for example, is very prone to encourage people to find out. I'm not. I'm just, you know, it just depends. Every individual is different, and again, it becomes an aspect of health disparity, because not necessarily if you're well prepared to deal with the information, you have access to it, because there is a cost associated to it. So it goes to healthcare inequalities as well, because it's not available to everyone. Yeah. Yes.
If I'm not mistaken, you said that the actual diagnosis is post-mortem, right? Is there not a way, like, to see those lesions before, like that? Are there like certain tests to find those?
Yes, and still you can find it in individuals that are completely cognitive normal, and it's the brain imaging. But again, these tests are only research only. They are not covered by health insurance, are very, very expensive. And that's why we're studying some of the projects that we're doing is in biomarkers, especially using having race and ethnicity as a variable, social variable, to look at finding that sensitivity that we need, because I'm sure, completely sure, that brain imaging will not be a standard care for people with dementia, because Medicare will collapse in a week. I mean, PET scanning, you're now, you have 6 million people, right? And imagine the people that are routinely checked, that are upcoming with my cognitive impairment, and you're gonna check them every year with PET scanning? I don't think so. The health insurance, Medicare, Medicaid will collapse. And the health insurance definitely are not covering that, because there's no treatment. So the idea is to find a marker that I'm convinced that is outside of A, beta, and tau. And we have some already in the pipeline. Hopefully we can validate. The problem is that I'm concerned that the ones that we're finding are in a specific cohort in the Michigan, in Ann Arbor and Grand Rapids area, Detroit area, right? And that biomarker can be for Michigan, but might not work for Texas or California or, you know, New York. So every state might need to, because of the social structure, every state might have their own biomarkers in order to get to diagnose, because I believe that is plasma what is going to take us to the early diagnosis. So let's say we have these biomarkers before my cognitive impairment, and we can detect it and say you are at risk. So Mediterranean diet, exercise, yoga, or, you know, meditate, or finding ways to have a multidisciplinary group of therapists, neuropsychologists that I intervene with you in order to prevent the clinical presentation will save a lot of money than when you have individuals with full-blown dementia. But we are not there yet. Yeah.
You just said right now Mediterranean diet, and does that actually have like a link to reduced Alzheimer's?
Yes.
And how, like, what's the, like, what aspects of the Mediterranean diet make it less?
Is the omega-3 and, you know, the lipids in fish that actually are better The wine with the antioxidants that are in wine, that they take one glass every day or things like that. They're antioxidant in wine, not a whole bottle. That's a problem. When you get the whole bottle, that's the problem. But a cup of wine, that is polyphenols. And what they're doing now is that, of course, they're putting polyphenols in pills. And you can have then, instead of the alcohol aspect, you can have the polyphenols concentrated, and those are antioxidants. And there is a relationship in between inflammatory response and the accumulation of radicals, oxygen radicals, and an oxidative stress in the cell. And then you accumulate those antioxidants, and then you have inflammatory response, and then that is a vicious cycle. So those aspects contribute to people that are in this Mediterranean diet have a lower risk. The question is, based on the theme of the presentation that I mentioned here, is the Mediterranean diet or the Mediterranean diet in the Mediterranean countries, right? So that's the other aspect that we have to keep in mind. But exercise, for example, is important. Sleep, the value of sleep. We are in a country that is completely sleep deprived. There are more accidents of people falling asleep in the road, car accidents, than people that are under alcohol or under drugs. So that is known. So what happens is when you sleep and you reach a REM, the blood-brain barrier and also the lymphatic system opens up, and it flushes out all those contaminants that you accumulate during the day. And that goes to the blood and then goes to the kidney, and then you eliminate those toxins. If you don't sleep, that stay accumulating in your brain. You don't sleep well, right? Or you have at least 6 hours for adults and 8 to 10 hours for younger individuals. So that, but it has to be 6 hours of a straight run, and what the studies done in animal models, and actually even in bears, when they go in hibernation, they actually, you see that they even inject A-beta, aggregate A-beta, and when they induce REM, they flush this out of the brain. And when they keep the mice awake, the A-beta stay in the brain. Yeah. So sleep, exercise, good diet, that will keep your brain in shape. Studying, reading, socializing, laughing, that always helps. So that's the information we bring to the community, that you don't have to go to a 5-star hotel to have a really good vacation in order to relax.
How would you promote this type of detecting Alzheimer's? Because I have a possible case in my family. The person does not want to realize any test because he says that there is nothing like he can do if it's detected, because it's just like a possible case. How would you promote people to actually go and get this test? My mom actually, she has a genetic disease, and she said that the only answer to it is if I exercise. So many people don't consider exercise and diet as a solution to problems. So how would you promote getting Alzheimer and other genetic conditions detected? Because I would like to be a genetic counselor. Yeah. And many people don't realize the effect genetic counselors have. And in Puerto Rico, there's not many genetic counselors, especially because Medicare doesn't cover it. So how would you promote people getting this type of test really?
Is one of the messages that we are into the community to try to go early detection, and it's a big problem. But if you want to go to the route of genetic counseling, you are right in the right timing, because it's not only in Puerto Rico, in the whole United States, there are shortage of genetic counselor. And what is happening is that the hospital system are providing that service. And then they work with the insurance or the out-of-pocket, or because what some health systems in the United States are considering and recruiting genetic counselors to reduce the burnout of physicians that have to deal with these situations, which create another economical problem for the health systems, right? So then they have this to refer, and in that way, eliminate the burnout and in physicians, especially neurologists. So to your point about your career choice, right on it, it's something that you're gonna find a job just like that. University of Michigan has one of the certified, accredited, I should say, genetic counselor schools of the nation. So if you want information, we can connect you there. Number two, let me put in perspective that inherited Alzheimer disease is less than 5% of all cases of Alzheimer disease. 95% plus of the cases don't have a family history. So even though we're talking about the inherited Alzheimer disease, it's very rare, okay? In your case, then to the big question, how you encourage people to go to get the diagnosis. That's something to talk with the entire family, to bring the doctor into the conversation, the physician into the conversation, how to manage the situation. Especially with you have a family medicine doctor or someone that regularly see this individual because of other reasons, engaging the physician in that discussion with the patient is something that you can do. So you can call the physician and say, hey, we're noticing this, and he or she is reluctant to go and get a check. I know that he or she has an appointment coming up. Can you bring it up? Can you talk about it? Because, the physicians come with another perceived power and can be influential in the decision. So involving the family doctor, the physician in that conversation will actually release the stress and the family problems that can be by bringing all the family to the table and talk to the person and all that. If you bring the physician, that will help, will have better outcomes. Yeah.
And you talk about APOE3 as in the case that the person showed the mutation in penicillin 1. Can that be a innovator in the future of that type of mutation in Puerto Ricans? Like, is that an outgoing investigation right now?
Well, the research right now is to try to find out why APOE3, epsilon 3, with that Chris mutation, what is the function and the intersect with APP pathology or presenilin, in terms of if it's regulating. Let me put it this way. Presenilin and presenilin 2 are proteases that are inserted in the membrane, okay? They cleave APP that is inserted in the membrane, and the enzymatic activity takes place in the lipid area, okay? So one of the ideas is that these APOE proteins are affecting the enzymatic activity within that lipid bilayer by inserting cholesterol or taking cholesterol out, changing the phospholipid composition. Something is happening, right? But those are ongoing studies. And the idea is that once you find that out, that's the next question. Is that applicable to all the presenilin 1 and all the APP? Is more general. Is protected to everything, or is protected to the combination of that presenilin 1 mutation with that Christchurch? We don't know.
We have to finish our
Okay,
presentation.
good.
Nevertheless, for the Zoom meeting, we thank Dr. Vega. Thank you, Dr. Vega.
You're welcome.